Hello everyone. Today we are diving into forensic biotechnology, a fascinating field where biology meets law enforcement. So, by definition, forensic biotechnology is the application of biological techniques and methods to solve number one, crimes, and number two, legal disputes. It's crucial in criminal investigations. It helps to identify suspects, exonerate the wrongly accused, and even solve historical mysteries. It includes DNA analysis, forensic microbiology, forensic toxicology, and many more. A great example is how the DNA evidence has been used to either convict criminals or free the innocent individuals. This scientific discipline integrates biological sciences with law enforcement, making it one of the very important tools in modern investigations. So if we look at overall, this field of forensic science, it has come a long way. In the late 19th century, fingerprint analysis was introduced as a method to identify the individuals, in their unique manner or in a unique manner. Then in 1953, James Watson and Francis Crick discovered the DNA double helix, which actually laid down the foundation for modern forensic genetics. And then in 1984, Sir Elect Jeffreys developed DNA fingerprinting, which revolutionized the field of forensic science. Today, with next generation sequencing and forensic bioinformatics, we can analyze even the smallest genetic material with remarkable accuracy. Over time, these advancements have significantly improved criminal investigations and justice processes. Forensic biotechnology uses a range of cutting edge techniques. Polymerase chain reaction, which allows us to amplify even a very small amount of DNA samples, even the minuscule amount of DNA samples. While short tandem repeats, which are also known as STR, the STR analysis helps to identify individuals based on unique DNA sequences. Mitochondrial DNA analysis is particularly useful for identifying remains in the degraded samples, which is often the case, when you have any kind of criminal case or cases like, you have murders, etc., where over a period of time, the samples actually degrade. Next is next generation sequencing, or the NGS, which provides detailed genetic profiling, which helps the forensic experts to analyze biological evidence with precision. And these methods make forensic investigations more reliable and conclusive. Variable number tandem repeats, or the VNTR, and short tandem repeats, or STR. They are both types of repetitive DNA sequences which are used in forensic biotechnology for DNA profiling. The VNTR consists of long repeat units, about 10 to 100 base pairs, while STRs have shorter repeat sequences, 2 to 6 base pairs, making STR more commonly used due to the higher resolution and easier amplification via PCR, or via polymerase chain reaction. Since the number of repeats which you can see here, varies significantly among individuals. Analyzing specific VNTR or STR loci can create a unique genetic fingerprint for identification in criminal investigations, paternity testing, and missing person cases. STR analysis, in particular, is the standard in forensic DNA analysis because it provides highly accurate and reliable individual identification. One of the most powerful tools in forensic biotechnology is DNA fingerprinting. The process includes DNA extraction, amplification via PCR or via polymerase chain reaction, electrophoresis, and profile analysis. This technique is widely used in crime scene investigations, paternity testing, and missing person identification. A well-known case where DNA evidence played a crucial role is the O.J. Simpson case. It's known as O.J. Simpson trial. The O.J. Simpson case was a landmark trial where DNA evidence linked Simpson to the murders of his ex-wife, Nicole Brown Simpson, and her friend, Ron Goldman. Where the blood samples were used, and the blood samples which were found at the crime scene, his car, and his home. Forensic microbiology. The definition of forensic microbiology is the study of microbes in forensic investigation. So, forensic microbiology involves the study of microbes to help in criminal investigations. Microbial signatures can help trace suspects and victims. This field is crucial in cases of bioterrorism, where bacterial or viral agents may be involved. A prime example is the 2001 anthrax attacks, where the forensic microbiology was instrumental in tracing the origin of the anthrax spores used. By analyzing microbial DNA, scientists can track outbreaks, identify bioweapons, and assist in solving crimes. Another field is forensic toxicology, which focuses on identifying toxins, poisons, and drugs in biological samples. Techniques like GCMS, which is gas chromatography, mass spectrometry, and immunoassays help detect substances in the body. This is crucial in cases involving especially drug overdose, poisoning cases, and substance abuse. One famous example is the Jonestown massacre, where cyanide poisoning led to mass fatalities. The Jonestown massacre, which refers to the tragic mass murder-suicide that took place on November 18th, 1978 in Jonestown, Guyana, where more than 900 members of the People's Temple led by Jim Jones died after being not forced but coerced, or convinced into drinking cyanide-laced flavor aid. So toxicology, is an essential component of forensic science, as it helps determine the cause of death and the presence of harmful substances in the biological samples, and especially belonging to the victims. So, what are the applications of forensic biotechnology in crime investigation? Forensic biotechnology plays a vital role in crime investigations, like number one, helping in identification of suspects by matching the DNA to crime scene evidence, helps to exonerate the innocent. Something which there is a project which is called Innocent Project, which has successfully has been successful in exonerating people who were accused of wrongful convictions. It helps us to identify disaster victims, such as the example is the victims of a 9/11 attack. We have DNA databases like CODIS, which are also instrumental in tracking the repeat offenders. So, what is CODIS? CODIS is Combined DNA Index System. It is a national DNA database. So, when I'm saying it is national, that means it is specific to US, and it is maintained by FBI, or Federal Bureau of Investigation, that helps the law enforcement agencies to store, search, and compare the DNA profiles to identify suspects, link crimes, and solve cold cases. And it consists of multiple levels. So you have the local, then you have the state level, and then you have the national database. And it includes DNA from the convicted offenders, arrestees, the people who have been arrested and they are under some jurisdictions, forensic crime scene samples, and missing persons. So CODIS contains profiles of approximately 16 million convicted offenders and arrestees and about 750,000 crime scenes. The STR loci used in the US Combined DNA Index System, which is the CODIS database, are scattered throughout the human genome. The original 13 are highlighted here. As you can see, they are in yellow. And then later on, these 7 more loci were added into January 2017, which are highlighted in green. So these 13 highlighted in yellow and 7 which are highlighted in green total are the ones which these are the loci which are actually used to identify and track different crime scenarios and criminals. These applications not only strengthen the legal cases, but it also ensures justice can be served with scientific accuracy. So CODIS uses 20 core STR loci for profiling individuals. The image here shows 4 electroferrogram panels, each showing peaks. You can see here, each showing peaks representing the STR alleles at different genetic loci. And each color represents a different fluorescent dye labeled primer set used for the multiplex PCR amplification of the STR loci. So the panel A here has got the blue peaks, which shows the loci, for example, D8S1 179, and each peak corresponds to an allele at a specific locus. Similarly, we have the second panel which shows the green peaks. These are other additional STR loci which are mentioned here. The third panel has got the black peaks. And then we have the fourth panel, which includes amylogenin, or AMEL. This is for the sex determination, and also for loci such as D5, S818, and FGA. So the ML peaks, X and Y, they indicate whether the sample is from a male or a female. So this is how you have these multiple loci which are then analyzed simultaneously, and that's the reason why we call it multiplexing. So each peak pair represents the alleles inherited from each parent. So homozygous will show one peak. So this is homozygous here. So one peak shows homozygous. Heterozygous means two peaks. So this is two peaks. So heterozygous means two peaks. And as I mentioned to you, the ML, which is the last one, this is the sex marker, whether, if it is XX, it is female, if it is XY, male. And this actually slide shows X and Y, so it's a male sample. So the presence and size of these peaks are used to create a DNA profile. Now let us look at some of the landmark cases which were where the forensic biotechnology has been used in order to solve the case. We have the Golden State Killer, where the killer was identified using forensic genealogy and DNA evidence. The Boston Strangler case, which was solved posthumously through DNA testing, confirming Albert DeSalvo's involvement. The Innocence Project case, where this project has, the project involved freeing numerous wrongfully convicted individuals using DNA testing. And the very famous Jack the Ripper case, which has seen recent forensic analysis attempts using mitochondrial DNA. These cases highlight how forensic biotechnology has transformed crime resolution. The Boston Strangler. The Boston Strangler case, which involved the murder of 13 women in 1960s, remained unsolved for many, many years, for decades actually, until the forensic DNA analysis which was done in 2013. So you can see the advancement of forensic DNA analysis to solve cases which were very old cases, something which was done in the crime which was done in 1960s. And actually, using the DNA analysis, the the investigating agencies were able to establish the link between Albert DeSalvo to the final victim, who was Mary Sullivan. And this whole thing happened through or using the preserved biological evidence. So the biological sample was preserved, and using that, the breakthrough happened. And actually, with this, the confirmation came that the Albert DeSalvo was involved in at least one of the murders, though a lot of people still doubt whether he was solely responsible for all the crimes, or did he commit only one crime, or there were more people involved. But overall, this case, in at least in one of the murders, Albert DeSalvo was convicted. Often we find that at the scene of crime, or the scene where the some criminal activity has taken place, often the biological samples that are collected, they degrade over a period of time. So this was a cause of concern, because then if the samples are already degraded, then it is difficult to carry out any investigations. uh, in order to address this issue, the mitochondrial DNA analysis entered into forensics. Mitochondrial DNA, which is around 16,569 base pairs circular genome, it is inherited maternally, and now it is being commonly used in forensic science to analyze degraded samples, especially like bones, which degrade over a period of time, hair, blood, especially in missing persons and unidentified remains cases. Since the late 1990s, mitochondrial DNA analysis has been peer reviewed, and it is now the evidence of mitochondrial DNA is now accepted in criminal courts, and people have recognized that it is a reliable method for forensic identification, even in extreme degradation cases. So, mitochondrial DNA analysis can also be used in, as an additional method to assist in identification efforts. So, we have the other evidences, and then if we want a little bit of more confirmation, then mitochondrial DNA analysis is also done. The Jack the Ripper case has seen recent forensic analysis using mitochondrial DNA from a shawl allegedly linked to one of the victims, Catherine Edovis. So, it was in 2019, researchers claimed a match between the DNA and the descendant of the suspect, Aaron Kominski, though the findings remain controversial due to potential contamination and the limitations of mitochondrial DNA. Despite its advantages, forensic biotechnology has its challenges. Number one is sample contamination, which can lead to false positives, which has, one can raise the concerns about the accuracy of the results. There are also privacy issues regarding the DNA databases. Should law enforcement have access to genetic information from the public sources or not? Additionally, the legal system must address the admissibility of forensic evidence in court. A key example is the controversy around familial DNA searches, which have led to both breakthroughs and wrongful convictions. And of course, we have the ethical considerations, which remain a major aspect of forensic biotechnology's evolution, and there are opposite views of stakeholders regarding the ethical aspects of using this technology in criminal investigation. So, those who oppose using the DNA analysis in forensic science, they quote this example where in 2004, Brandon Mayfield, who was a US lawyer, he was wrongly arrested for the Madrid train bombings after the FBI mistakenly matched his fingerprint to evidence found at the scene. Early DNA analysis also suggested a possible link, but further testing by Spanish authorities proved the DNA did not belong to him, leading to his, overall release after 2 weeks in custody. The case highlighted the dangers of, forensic errors, particularly in fingerprint and DNA analysis. And actually, this case led to a $2 million settlement for Mayfield from the US government because obviously he's a lawyer, so he understood his case, and then he actually became a recipient of $2 million for the settlement of his case. So, in order to perform the DNA profiling, the forensic scientists they follow a structured process in the lab. Number one is sample collection, where DNA is obtained from sources or the biological specimens. For example, from it could be blood, saliva, hair, skin cells found at a crime scene. Next, DNA extraction. So DNA is extracted from these biological specimens. The quantification is done where the amount of extracted DNA is measured to ensure that there is enough material for analysis. Then the PCR amplification. So the specific regions of the DNA, they are copied multiple times using the polymerase chain reaction technique. Then separation by electrophoresis. These DNA fragments are separated based on the size using agarose gel electrophoresis or capillary electrophoresis. The STR analysis, which is the short tandem repeats, they are examined to create a unique genetic profile. And then the comparison and interpretation is done where the DNA profile which is generated is compared with either the known samples in the forensic database or the CODIS, or from different suspects or the family members or the neighborhood, if there are public databases also available, which is the IgG. And then we have the final report and conclusion, where a detailed report is prepared, which can be used as an evidence in legal cases. So, how do we interpret the DNA profiling results in criminal investigations? So, once the DNA profile, has been generated, the forensic experts can use the results in many steps or following many steps. Number one, matching profiles. So, the DNA profile which is obtained from the crime scene is compared with the profiles from known individuals, including suspects, and we can also use the database records in order to identify the perpetrator or the criminal. Next is probability calculations. The likelihood of a match occurring by chance is calculated using statistical methods. So a low probability indicates a strong match. Then we have the exclusion or the inclusion step. If the suspect's DNA profile does not match the crime scene evidence, they are excluded as a contributor. But if it matches, they are included as a potential suspect or potential source. Mixture analysis. In cases where you have multiple individuals contributed the DNA, the forensic analysts they separate and identify the individual profiles. Contamination checks. We need to ensure that there is no accidental contamination, while handling the biological samples, or we need to avoid any other environmental factor which can lead to the contamination of the source of DNA. Next is legal considerations. The evidence that we have collected is that ultimately it needs to be presented to the court. So when the evidence is presented in the court, it needs to be accompanied by an expert testimony explaining the results and their significance. So there, somebody who understands the DNA profiling, somebody who has an understanding about the mitochondrial DNA, or the use and the use of CODIS and STR and VNTR needs to be present there in order to convince the judicial officials about the validity of the results. So around the world, including Philippines, has actually realized the importance of forensic biotechnology. So the Philippines plans to increase its pool of forensic experts and the medico-legal investigators by bringing in experts from Australia who will help the University of the Philippines offer educational and training programs in forensic science. So in July 2024, the DOST, Philippines Council for Industry, Energy and Emerging Technology Research and Development, inaugurated the Forensic Molecular Biology Center. And this is situated at Sultan Kudrat State University. And this facility is valued at 4.96 million pesos. And this is the first of its kind in Mindanao. And this is going to help us to train a lot of people in this field, which is going to be used. These experts are going to be very helpful in bringing down the or helping in the law and order of the country. So how does the future of biotechnology look like? The future of forensic biotechnology looks very promising. Artificial intelligence is being integrated into forensic science, automating DNA analysis and crime scene reconstruction. CRISPR technology may have potential applications in forensic science very soon. Rapid DNA testing devices are being developed for instant suspect identification. A fascinating advancement is the DNA phenotyping, which creates physical suspect profiles from the DNA samples. These innovations are going to make forensic investigations much faster, more accurate, and much more efficient in the years to come. So with that, we come to the end of this presentation. I hope you got an insight into how biology is shaping the future of crime investigation. Thank you so much for listening. These are some of the references which were used in this presentation. This is a fun exercise. You can try to find out who did the crime by looking at the data given to you. And here is the answer for the previous slide. With this, we come to the end of this presentation. Thank you so much for listening. For more information on this topic, please visit www.biotechnologyforyou.com.